Hi everyone, thanks Bill for the introduction. I hope you can hear me, and I'm going to share my screen so assume you can see my slides. So welcome to everybody to this symposium. I hadn't heard about it until I was asked, and I think it's a great opportunity for promoting bioinformatics among our youth. So I'm going to give you a journey of my career, but really as it applies to what bioinformatics has been past, present, and future. Most of it obviously focusing on the present, because that wasn't always there in the past, and we don't yet know about the future, but we can predict. So I'm going to talk a bit about where and how I started in the field. Obviously, through this time, there's been a big move to big data, and the data is getting bigger and bigger. So how did we get there? And what is the value of this data? And some of the bioinformatics challenges. So why is bioinformatics so important in the interpretation of life science data? And then I'm going to talk quite a bit about how bioinformatics is being applied in human health, because that's sort of the sector that I work in. A quick peek into what I think the future might look like, and generally how computational biology and bioinformatics is changing our world. So I'm currently a professor in bioinformatics, and as Bill said, I head the Computational Biology Division at the University of Cape Town. This is a picture of it. This is in fact a picture of the main campus, and I work on the medical school. And I also run, as Bill said, Bionet. So this is a Pan-African bioinformatics network of more than 30 institutions across the African continent. And the aim of this network is to build capacity for bioinformatics research in Africa. And that's to enable people to analyze genomic data for health. I also co-lead a sickle cell disease data coordinating center. So sickle cell disease is a very debilitating disease caused by a single mutation in the hemoglobin gene. And it's predominantly found in people of African descent, so people with African ancestry. So there's a big consortium across Africa to manage sickle cell disease, and we manage the data coming out of that consortium. So where did it all start? Well, things don't quite go according to plan, and my plan was to be an artist and to study art and graphic design. But then in my final year of school, I went to the guidance counselor, as we all have to do. And she said, oh, well, with your marks, you could do a degree at university. So I said, oh, what should I do? So she showed me the BSc. I said, it looks really cool. The subjects look like fun. So I registered, and I ended up doing BSc, a Bachelor of Science in Microbiology and Chemistry. I enjoyed the microbiology side, so I went on to do honors in that, and then finally ended up with a PhD in medical microbiology. So somewhat change of course from my original plan. So PhD work was a lot of, it's sort of, towards the end of it, I got into a bit of bioinformatics, but the aim of the PhD was really to look at transcription factors in an organism called Microbiomes tuberculosis. So this is a bacterium that causes TB, tuberculosis, and TB is known to have different disease severities and outcomes. So when you get infected, some people go onto active disease, while others go into latent infection. And this can be reactivated later in life or with other comorbidities such as HIV. So my PhD was trying to look at how Microbiomes tuberculosis, this bug, is able to move from the active state, so actively growing, to a kind of dormant or state of hibernation where it sits in granulomas or in the lungs and then can be reactivated, because in that state it's not susceptible to anti-tuberculosis drugs. So at the time, the genome sequence, the DNA encoding the instructions for this bug was not yet available, so I had to go through other organisms that did have a genome available and try to find genes that might be involved in this latent state, and use a process called PCR, polymerase chain reaction, which is a lab technique for amplifying something specific out of a genome. That's in fact what they use for the COVID-19 diagnostics. And use a process called Southern blotting to see, well, are these genes in another organism also found in the bug that I'm working on, tuberculosis? TB was in fact very difficult to work on because it takes 14 days to grow as opposed to a few hours for E. coli. And of course, it's highly pathogenic, so you have to work in safety level laboratories. That was another reason why I moved to bioinformatics. Anyway, halfway through my PhD, the TB genome was sequenced and all that work that I'd started on could have been saved. But anyway, I then carried on and worked specifically on these genes in Microbiomes. So I ended up having to do some sequencing and some bioinformatics, which was all self-taught at that point because we didn't have bioinformatics in South Africa. So how did we do this then? Well, at the risk of showing my age, I did my own sequencing in the lab, running sequencing gels, these plate glass gels with radioactively labeled materials. So you had to work behind lead sheets. And then the image on the right is the kind of picture that came out with these lines showing where's the A's, T's, G's, and C's. And you literally went with a ruler and wrote it out. You can see the potential for human error. And then type this out. So that was my sequencing experience. The internet had only just emerged. So when I then wanted to run my sequences against public databases, so one of the big applications of bioinformatics is to compare things to what exists. So I had to run the sequences against these big databases of existing DNA sequences. At that point, we didn't have online blasts, so we had to get these disks from these databases shipped to us, put them in our desktop and run them directly off these disks, which of course sounds to you completely archaic. But at that point, that was all we had. And then we just had various DOS-based interfaces and no HPCs. We had to do everything on these little old desktops with our disks. So that's how we did things then. When I finished my PhD, I then traveled for a year in a camper van through the US, Mexico, and then a separate one through Europe, which was great fun. But then finally I decided it was time to get back to real life and take up a job. So I took up a position at the European Bioinformatics Institute. This is in Cambridge in the UK. And there I worked as a curator for a database called SwissProt. So this is a protein database that's highly curated. So you have biologists coming in and annotating these proteins based on the literature, so based on experiments that people have done. I didn't stay in that for very long because a new project came along, but I'll come back to that. So during my time at EBI, which is a part of the European Molecular Biology Laboratories across Europe, we did, so I did some work as a SwissProt curator, and I did a lot of bioinformatics courses there. The Human Genome Mapping project was based on the same campus. So this is the campus where the Sanger Center is. And so I did some bioinformatics courses there. And then I got involved in a new project to functionally curate, functionally annotate proteins in terms of their families and domains. It's called Interpro, and I eventually became leader of that project. Now, just what was really quite a good opportunity at the time was that I wanted to, as a biologist, I like to put things in boxes. So I wanted to be able to, thinking back to my PhD, I thought, now if I had to run Microbiomes tuberculosis, the genome through Interpro, can I create a beautiful pie chart to say this set of proteins is involved in this kind of function, this kind of proteins are, this set is involved in this. So basically divide the genome up into its different functions. And there was no easy way to do that. So I developed my own sort of functional classification scheme. And almost in parallel, just after that, a new scheme was being developed called the gene ontology. But that wasn't available yet at the time. So I developed this functional classification scheme and mapped it to Interpro entries so that if a protein belonged to an Interpro entry, it had a particular function. Somebody heard about this, and at the time, people at EBI and across the corridor at Sanger were involved in this sequencing of the human genome. And they needed a functional classification of this human genome sequence, this new sequence. So I got involved and I applied my functional classification scheme to this and created some of the figures for the Nature paper on this human genome. So looking at the human genome work, which is pretty landmark, I think, in biology and in bioinformatics. We think about the fact that in 1953, the structure of DNA was discovered by James Watson and Francis Crick. It took nearly 50 years to sequence the human genome for that to, well, for the human genome to actually be released. So this was released early in 2001, and it was actually a race between the public and the private sector. So the Nature paper was this public group of academics, International Human Genome Consortium, and the science paper came out from a company called Celera, where they were trying to sequence it and potentially patent some of the genes. And it just wasn't right to have the sequences owned by somebody. So they put pressure on the public effort, and eventually the two and now these two issues came out in the same week. So just some statistics about that genome. So we have about 3 billion bases. 99.9% of our genomes are identical. A portion of our genomes are our own. So when we've been infected in the past with viruses, some of those viruses tend to insert pieces of their own genome into ours. And so you've got relics of viral origin that are sitting in our genomes. It was a surprise that there was a poll that went out, sort of almost like a ballot to say, how many genes do we have? And there was anything from a few thousand to hundreds of thousands. But actually it turned out that we only have about 20,000 genes, which was a bit surprising considering that some very simple creatures contain the same, if not more. And the rest of the genome was considered to be junk DNA. So what did it code for? It turns out now that it's actually not junk. It obviously wouldn't have a huge 90% or 98% of our genome coding nothing. So these were found to be other kinds of genes, non-coding genes, pseudogenes, other kinds of non-coding pieces of sequence that had a particular function as in themselves. A pseudogene, by the way, is a gene which used to have a function, but it slowly gathered so many changes to it that it's no longer a functional protein. So just some trivial information about our genome. If you unwound it and tied together the strands of the DNA in one cell, one of our cells, it would stretch almost 6 feet, but it would only be 50 trillionths of an inch wide. If all the DNA in our body was put end to end, it would reach to the sun and back over 600 times. So that's how much DNA we have compacted into all the billions of cells we have. It would take a person typing 60 words per minute, 8 hours a day, around 50 years to type the human genome. So it's quite lucky that the data can be captured automatically now. And if 3 billion of all 3 billion letters were stacked 1 millimeter apart, they'd reach a height 7,000 times the height of the Empire State Building. So it's much bigger and more complex than I think most people think, even though it's small and you can't even see it by eye. So one of the biggest things that came out of the genome was, the promises were the genome's going to just answer all our questions about health and humans. What did it tell us though? Actually not much. The genome did not tell us that much. At the time we knew that Down syndrome was because of an extra copy of chromosome 21. It still didn't tell us why. Or how that causes a particular phenotype. And it's because most non-communicable diseases have a strong genetic component. They also have an environmental component. Infectious diseases are affected by both the host pathogen and then also the environment. But luckily, since then, a lot of genetics research has been done that has been enabled by having the genome available. And this has enabled us to find additional links between the genome and diseases. That's been invaluable, and people have been able to act on it and save lives. So interestingly, there's the sequencing, the human genome sequencing project cost $3 billion US, and it took 5 years to complete. Now you can sequence a human genome for $1,500, some places even less than that, for less than a week. So now you can generate human genomes at an unprecedented scale and for an unprecedented cost. But this does not consider the cost and time for the data analysis. So if you look at graphs for the typical graphs of how decreasing costs of sequencing compared to Moore's law. So Moore's law is the law by which prediction of it's the prediction of dropping costs of computing. And you can see that the dropping cost of generating sequence data is just at a different rate. And the sudden drop is when people were doing traditional Sanger sequencing, even after I finished my plate gel sequencing, they got more advanced and they did capillary sequencing. But after that, in about 2008, you got next generation sequencing, which was suddenly massively parallel. So instead of having kind of one experiment on the go at the time, these experiments could be done in parallel, and you suddenly got billions and billions of pieces of sequence for a particular genome that you were sequencing. But they were much smaller pieces than you would get with traditional sequencing. So this came with bigger data, but more complexity and more fancy algorithms to analyze this data. And then, as I said, the sequencing component is becoming smaller. So at first, pre-NGS, the blue in the graph here, shows that most of an experiment was about the actual sequencing. And then the peachy orange color at the bottom was how much time was dedicated to, and the cost was dedicated to downstream analysis. As we get more and more genomes and we get more and more data, sequencing costs are coming down, and the costs of the actual data analysis are increasing exponentially. And this has led to, so the decrease in cost of sequences led to a huge increase in the amount of biological data. You can see the scale here. And people were predicting that just from sequencing alone, there will be an 100 times increase in the amount of data. I think it's going to be even more than that. But we're getting more and more data of different types. So there's sequence data, there's array data, there's all the other robotics that are coming in to create different kinds of data. And this means new algorithms needed. So how did we move as little biologists working on single genes? I worked on 3 different genes in my PhD, to working on such a big scale. When I was working, I could manage on a really old desktop. Now you couldn't. Many bioinformatics techniques require high performance computing clusters. And the reason is that we've moved to massively parallel generation of data. So we have everything's done by robots these days. We get billions of data points from sequencing, large-scale genotyping data. Mass spectrometers are producing huge proteomics data. So genotyping data is data where you're measuring a change in the DNA sequence. Proteomics data is when you're seeing what proteins are in a particular sample. So all of this has resulted in the need for greater processing power, greater storage capacity, and also data interpretation, because the data is much more complex, coming from all these different sources, and we need to be able to put it together to answer the biological questions. So if you, for those of you who don't know, what is big data in the first place? Well, it's usually classified by the 3 V's, volume, velocity, and variety. So that's how much data is coming at any one time. For biology, for life sciences, volume is not, for us it sounds huge, but it's not as big as, say, the rate at which astronomy data is being collected, for example, or some other data types. Velocity, so the data speed, again, the telescopes can generate data much faster than we do in the lab, but the speed has come, because of these robotics, the speed has increased. Then you have the variety, and this is the biggest problem, I think, with bioinformatics is the variety of different data types in biology is just huge. And so we're dealing with very heterogeneous data sets, trying to pull them all together. And then you could add, it's not one of the 3 V's, but you could add veracity as well, so the accuracy of the data. How accurate are these, as we generate data faster, is that increasing the error rate at which we generate data? So just some stats on comparing different data types. So apparently Facebook ingests 10 terabytes of new data every day. Twitter generates 7 terabytes of data every day, and this has probably even increased since I wrote these slides. Genomics databases currently are several petabytes in size. When I worked at the EBI, probably 10 years ago, they hadn't even got the first petabyte yet. Now we're in several petabytes. So that's how it's changed. It's an estimated that overall data, all kinds of data, that 2.5 times 10 to the 18 bytes are generated daily. And IBM has predicted that 90% of the current data that we have stored was generated in the last 2 years. So we've generated more data in the last 2 years than we've generated almost in many people's lifetimes. And the issue is that most data is generated by machines now. So it means that it's sometimes unstructured. It wasn't really designed for user or computer friendliness. So there's a lot of processing that needs to happen to make it useful. But we have to make it useful because there is so much value in the data, particularly in the life sciences. I have this argument with the astronomers and other people to say whose data is the biggest and whose data is the best. And so what if you get to see a new planet or a new star? That's fantastic, but are you saving lives on the planet, on our planet? So my argument is that the life science data is some of the more valuable data. So we can make discoveries in healthcare and agriculture and food security as new genomes. And my focus, as you might have noticed, is on genomics, because that's really what I do quite a lot. More than 1.2 million species of plants and animals have had their genome sequenced. And so we have a lot of scope for discovering more about them to be able to increase our food security, to learn more about agriculture and health. Obviously, it's pretty topical at the moment, is disease surveillance is important. So this is data from the virus, from transmission networks, from host, for contact tracing. The data is all essential for stopping for curbing the spread of this virus. There's data that's coming in from electronic health records, so people visit doctors every day, not every day. Every day doctors see patients and they record what's happened to them. That data can help to inform their future health by what's something called precision medicine, which I'll come back to. Data, once analyzed and interpreted, can therefore benefit health. But unfortunately, there are many challenges. I've mentioned some of them, but I'm going to unfortunately dwell on a few of them now. So it was reported that in the US healthcare system, it's expected to reach the zettabyte, that's 10 to the 21 bytes, scale from electronic health records, scientific instruments, clinical support, decision support systems. So that's just in the US healthcare system. And how do you convert that into saving people's lives? The problem is that the data is coming in faster than we can process it, but also that people who are in the field, so clinicians, basic scientists, wet lab scientists, they weren't built, designed, trained to manage big data and to generate great algorithms. So you're kind of having to have a merger of two ecosystems here. And the other problem is that we've moved from the little pie chart on the left, where we had, in the past, when I did my PhD, we had a little bit of data, but lots of information. So we had data, but we knew a lot about that data because it was, switchboard, we were hand curating things. Now we have loads of data, but not that much information about each of those data points. So we're moving from data poor, information rich, to data rich, information poor. And what's the point in having data if it's not going to lead to information and knowledge? So we need to convert billions of data points into statistically powered evidence for some particular application. So if you look at this graph, this image at the bottom here, we have our genes that are encoded in our DNA. These genes code for proteins, so they need to be expressed in certain times and certain tissues and under certain conditions to make the right proteins at the right time. They have a particular function, they work in particular pathways, they adapt to particular environments to cause something, either a particular phenotype. So a phenotype is a characteristic of something. A phenotype for human would be their characteristics, either disease characteristic or blonde hair, blue eyes, whatever. And this is all encoded in our genes, which basically are the instructions for this whole process. So if we gather data points on all of these, how are we going to manage them? So some of the challenges we have are from these three V's. is how do we capture the data off machines in the first place, of the experiments that we're conducting that generate data. Once we've got it, we need to move it to the site where it's going to be analyzed. That used to be trivial. Now we're moving big data points. We tried to move 350 terabytes. It took us 3 months because of the internet challenges in Africa. Data storage. How many researchers have budgeted for data storage? Not many. Data analysis, processing, integration, querying public databases, visualizing data. It's all a major challenge. But one of the biggest challenges, I think, is for life science data, I think, is the heterogeneity and noise. So what is real? What is error? What is experimental error? What is just natural variation? Data comes in different forms, so binary, time series, single point intensity, images, values. And how do you convert that into something you can work with to compare against and come up with something useful and applicable at the end? Data has gaps. So not every experiment works. And so some points don't light up, some tubes don't work as they should. So you have gaps. The data's noisy. Information, the metadata, this is information about the data. So explaining what is in this tube versus that tube, how was the sample collected, all that is often poor. And it's in millions of different files and formats, particularly in bioinformatics. So it's important to have good visualization. So how do you sort of interpret data? As a biologist, I like to see things. I like to visualize pathways, patterns, so I can pick up what looks right and what looks different. And so visualization is a whole field of bioinformatics that's essential, because just visualizing something a different way can actually give you the biological answer that you couldn't see in a different visualization. So generally, the bioinformatics needs in this high throughput life science data is, well, we need more bioinformaticians. So we have more data, so we need more qualified people who can manage data. We need more data repositories and computing infrastructure. The data need to be harmonized. So this means that if data is coming from 3 different labs and they did similar experiments and you want to analyze them together, you need to make sure that the parameters were the same, that they collected the samples the same, that the data's curated the same way and described the same way. We need manipulation tools, visualization tools, prediction tools, decision support tools. And the problem is, as I said, that biomedical scientists are not necessarily good developers. Many of the bioinformaticians I know are wet lab people turned data people, which means that they didn't have formal training in computer science. So they'll learn from some online courses, they'll write some code, it's very buggy, it's not optimized, it's not scalable. I mean, that's very generalized, but people who are just working on their own tools, and so they're not the kind of optimal people to be developing new software. They can do data analysis, but if you want good unbuggy bioinformatics tools, that should really be done in theory by professionals. And the algorithms are constantly evolving and changing because the data types keep changing. I mean, you buy a sequencing machine, and a month later there's a new one that comes out, and then yours is out of date already, and then the data types might be different. So that's a general kind of list of challenges and what's needed for bioinformatics. But if we're working in human health specifically, there are additional challenges. So the data generation is expensive. It's somebody's personal sample that they maybe had a needle prick to get. They had to come into the clinic, they had to answer questions. So it cost them time and effort. It costs somebody a lot of money to generate the data, sequence data, for example, from that. The data is getting very big to both store and process. You have to combine it with image data. So, for example, you might have sequence data combined with an MRI. How do you process that and compare them? We have differences in populations, so different people, the genetic variations between different populations. So European populations are very different to African populations. You just can't compare them when it comes to health. And so you're looking at almost different species in terms of, the kind of data that you're looking at just by looking at different population groups. You have data access limitations. Suddenly when you're dealing with human data, it's personal, it's private. and you have to get ethics consent, and you have to obey the new laws like IP laws. There's the new GDPR and in South Africa prepared. So these are data protection acts for individual protection of private information. And in dealing with all of those challenges, you then need to also have the tools to translate all of the relevant data into something that's going to save lives. Coming from Africa, I talk about the specific challenges here. So this is, we have unique fauna and flora, so, and then also African populations are quite different, and yet hugely underrepresented in public databases. So if you look at public databases, it's moved up to maybe 8% of entire public databases are African relevant. And so we have to generate our own data. We don't have anything to compare against. We generally have poor internet access because we just poorly resourced countries, so which makes data transfer very challenging. It makes data processing very challenging. Inadequate IT infrastructure at universities, inadequate electricity, so you start to run a big job on a high-performance computing cluster, and then the government decides to cut the power for 3 hours. So you have to manage that, start again. We also have limited long-term secure storage, and generally just limited number of people who are trained at different levels to manage data challenges in Africa. And I mean, these are generic, that metadata is not well curated and that researchers usually don't budget for data, but we simply do not have enough bioinformaticians or data scientists on the continent. So that brings me back to my career journey. So I was in the UK. I am South African, so I was always planning to return home to Africa. And meanwhile, while I was at the EBI, the South African government put a whole lot of money into developing bioinformatics. So they developed this national bioinformatics network. So this is because I recognized the need for more bioinformaticians to start working on national projects. So I came back for a while. I was kind of commuting. I know it's a long commute, 12,000 kilometers from Cape Town to Cambridge, but I did it for about 3 years. I was working half at firstly at SANBI, which is South African National Bioinformatics Institute, and then moved to UCT, University of Cape Town, still working for the EBI half at that time. So I got to get my fingers dirty in Africa, but keep the fantastic ties and networking that I had and opportunities I had at the EBI. So my first year back in Cape Town, I got involved in the African Society for Bioinformatics and Computational Biology, which is, I guess, African equivalent of ISCB. And we started running conferences every 2 years for African bioinformaticians. And after our second conference, after the first conference, for the second conference, I got chatting to Manuel Corpus, who was, I think at that point, leading the student council of ISCB. And he said, hey, shouldn't we team up? So I thought, well, that sounds great. And we got ISCB board of directors on board. And we started teaming up to run what's called the ISCB Africa conferences. So these run now every 2 years. These are just some of the banners from some of those conferences. We've had them in Mali, Ghana, Tanzania, Cape Town, Uganda. And these have been really successful thanks to the teaming up with ISCB and sort of enabling African researchers and students to have access to excellent bioinformatics research and conferences. And these are always tied to some sort of training as well. So I got very involved in that. When I was at UCT, I started building up my research group. I was a National Bioinformatics Network node manager there. And I think a seminal moment was in around 2008 when I was invited to a meeting in Lagos, Nigeria that was organized by the World Health Organization, because they recognized that bioinformatics was important in Africa and that we should start to work on this. So at this WHO meeting, we had a few people from around the continent come together and start discussing the potential to build an African bioinformatics network. Had fantastic ideas, went away all enthusiastic until the WHO said, great work, guys, but sorry, we can't fund it. So there was that. But then in around 2011, the NIH and Wellcome Trust had come together through the African Society for Human Genetics and decided that genomics was important and that bioinformatics was important. And they actually put out a call for a network, continent-wide African bioinformatics network. And so we applied and I was successful, and that's H3A Bionet. So this is what I lead. So that was funded from, it's been funded since 2012. We're in our second round of funding now. So it's basically a Pan-African bioinformatics network to build bioinformatics capacity for a particular consortium. This is the Human Hereditary and Health in Africa Consortium, which was designed to look at, to fund studies in the genetic and environmental basis for diseases in Africa. So you can see in the map here, the different countries that we cover. The tiny dot in the ocean is Mauritius. That's one of our nodes there. So we have 28 partners, although that kind of increases up to about 34 at some stages, more than 200 different members. We do a lot of capacity development, skills development, research, bioinformatics research, tool development, and a lot of training. So we run a big online remote course where we're currently training 1,000 people across the continent all at once. And we've trained over 2,000 people to date. But the idea of this has been to facilitate the study of human diseases in Africa. So that brings me on to kind of where I think bioinformatics plays a really important role. If we think that the human diseases is one of the key millennium development goals. And this is also a key part of my research. When I came back to UCT, I did quite a lot of work on TB at first. I was really into my bugs, bacteria and little things, because they're easy to work with. You don't have to get ethics consent from a bacterium to sequence it and work with it. But then through this, I moved on to human diseases and took on all the challenges that came with. So where does bioinformatics fit in? Well, if you think about the kind of the overall move in our cell from the chromosomes at the top, the individual sequence, how that is transcribed into genes. Genes make proteins. Proteins have functional domains. That's it. Where project is involved in. Proteins have structures. They work in pathways to make an organism work. So you can use bioinformatics to study the genome, the different features, the transcriptome, which is the expression of the genes to RNA, proteins, protein families, interactions. You can work it. Bioinformatics comes into every single one of these pieces of the whole system. And then specifically in health, as I said, it's a key millennium goal, and so it's clearly important. It's important because, infectious diseases and non-communicable diseases. So infectious diseases killed more people than the 2 world wars over the same period of time. And yet they kind of weren't reported quite the same. And as we all know now, with our little friend on the right, we have this emergence of new viruses, new bacteria, drug resistance and extremely drug resistant strains. But I think what's on everyone's mind right now is this emergence of this novel coronavirus, which I've never seen anything impacting our lives like this has. All the other, emergence of the Ebola and all of that, it hit a few people, it hit a few countries. Now we've hit the world. So this is something kind of unprecedented. And I'll talk later about how bioinformatics is helping there. Genetic diversity is really important. So this is both genetic diversity of the pathogen themselves. So the virus, the bacterium, their genomes differ, and their genomes, the differences in these genomes change how virulent they are, how likely they are to kill somebody, how strongly they elicit a disease response in somebody. And then there's the genetics of the host. Different changes in our genome can predispose us to different diseases, both communicable, which is infectious diseases, so how we respond when we get HIV infected, how we respond when we get infected by COVID, and then also the non-communicable or the lifestyle diseases like diabetes, cardiovascular diseases, heart diseases, all the ones that kind of are not from an infection, they're from a kind of lifestyle and genetics. And so omics, which is kind of the word that covers all those kind of techniques that I said. You can study the genome to give you genomics. You can study the transcriptome, so that's how genes are expressed. That's transcriptomics. You can study proteins and the whole set of proteins, which is proteomics. And studying all those different components of a biological system, they're all important in human diseases. It's not just about the genetics. Sometimes things only happen at the protein level, so we need to study all of these. And bioinformatics is a key interpreter of omics data. So if we're studying human health, talking particularly here from a genetics and genomics perspective, we have your pathogen. In this case, it's a bunch of TB, Microbacterium tuberculosis, and we have our host. From the pathogen side, we can use bioinformatics to look at pathogen surveillance and epidemiology, sequence the genome to look at drug resistance mutations, which changes are causing something to be more virulent, so causing worse disease. And then we can look at, we can model the transmission. So obviously for COVID, they're doing a lot of modeling of transmission. So where did it come from? You know, where was the virus picked up? Was it picked up from taxis, from schools, from homes, from public places? And we've done a lot of modeling of TB transmission in our local communities because it's quite an epidemic here. You can also then look at the host and the pathogen together. So what pieces of the virus or bacteria are interacting with what pieces of the host? And then for the host itself, there's all the analysis you can do on population genetics, which is basically an analysis of the differences between individuals. I said that 99.9% of our genomes are the same. It's looking at that little percentage that's not the same, and seeing how that is linked to diseases. And then, of course, there's the microbiome. So I don't know who's heard of microbiomes. So metagenomics is basically the study of everything in a particular community. So somebody can get a puddle of water, mush it all up, get the DNA out of it and sequence it. In doing so, you'll then be able to see, determine what organisms were in there. So the microbiome is just like a component of this, is what is the community of small things living within us in different parts of our body. So it's basically the human microbiome is a combined genetic material. So this is the combined DNA of everything that lives in a particular part of our body. So unfortunately, the number of microbes in our body outnumbers our own cells by 10 to 1. So as Stuart Brand says, if you don't like bacteria, you're on the wrong planet and you couldn't like your own bodies. By weight, we are more human than bacteria because your cells are bigger, but the numbers are opposite. So say the number of bacterial cells outnumbers ours by 10 to 1. And the interesting thing is that microbiomes are coming out as key indicators and key modifiers of health. So subtle changes in our microbiome can cause a disease. And then if you restore that balance, you restore your natural flora again, you can potentially get cures. So this is opening a whole new field of health research. So this has been done through something called the Human Microbiome Project. So this is where people have been studying microbiomes in different people and different body parts. And this microbiome changes with age, puberty, diet, medical conditions. As soon as you take antibiotics, you kill off your microbiome. And then you've got to, that's why they say take some probiotics to restore your microbiome. And there's also been a lot of links to diseases. So different parts, so you have some skin microbiome that could lead to psoriasis or acne. Some microbiomes of the gut can lead to irritable bowel syndrome, diabetes, obesity, etc. And the list of microbes that are in different body parts are quite different. So you'll see in the plot on the left, don't worry what the plot's showing, it's just showing that the clusters are the different types of bacteria or microbiome components in different parts of your body. So if you take some bacteria off your skin, it's not going to be the same as the bacteria you'll get from your stool samples, for example. So the microbiome has become a really interesting area of research, and again, without bioinformatics, you can't do microbiome research because it's analyzing genomes, it's analyzing sequence data, it's bringing together statistics, putting that together with clinical data to see how the microbiome affects diseases. So then back onto our kind of overall overview of genetics and genomics, I'll just move now onto population genetics. So this is something that I spend most of my life doing at the moment in my group days, is that's looking at genetic changes and how these changes are impacting health. So there are many different kinds of changes you can have in your DNA and your genome. So a single change where one base, one letter has changed to a different one. This can have implications, or it might be silent, as in it didn't, well, the genome's not bothered about it, it didn't change anything. Or it could change actually, it could be in a gene, it could change the protein, which can immediately cause a disease. Just one little change in your genome. But there's a lot of natural changes. So on average, there are 3 million differences between any individuals at the single nucleotide level. So if I'm sitting next to somebody, I can look at them and go, well, 3 million pieces of your DNA are different to 3 million letters in my DNA. And maybe that's why you look different, but also might impact how our health is going to turn out in future years. We can identify these differences from sequencing or by a technique called genotyping arrays. And then we can look at individual level differences, but we can also look at the differences at population level. So look at all people in the UK, look at all people in indigenous populations. So look at all people who live in Ghana from a particular ethnic group, compare them to another group nearby. So we can calculate what's called frequency. So how often do we see this change in that particular population compared to that change in another population in that same position in the genome? And we can start to do analyses on these. So this is just an interesting analysis where somebody looked at a whole lot of, this is a reasonably old paper, a whole lot of genomes from people from Europe. And when you split out the differences using what's called PCA, so principal component analysis, this just sort of separates out based on these changes you're seeing at the genetic level. It almost kind of maps back onto where the map is, the geographical map. So looking at people's changes can actually almost help to identify where they came from. So where they live, what is their ancestry, what is their population ancestry. But the main thing is that these changes have impact on health. So change in the DNA can cause a change in which proteins get made. If the protein is different, that can have a change in the final pathway it's being involved in, and therefore the final phenotype or the clinical characteristic. So we can study this at different levels. These are the different omics that I was talking about, the genes, the gene expression, or the transcriptomics, the proteomics, metabolomics, lipidomics, anything with omics. You can just throw omics onto the end of any word, and it's part of the omics. And what's interesting now is that we're combining this genetic variation and other omics data with other data. So we can get populations. We can say, right, we've got a whole population that's similar. Some of them are getting the disease and some of them aren't. So can we compare the differences in their genomes between these two to see, over and above all that natural 3 million natural pieces of variation, is there something that's statistically significantly different between them? And identify changes that are linked to disease. We then match this to perhaps electronic health records. So when you visit your doctor, they keep a record of everything. Can we match your genetic changes to what they've recorded? Add environmental data. So what have you been exposed to? Were you exposed to pollution? What is your diet? Other lifestyle factors? Wearable devices. So how often do you exercise? Have you measured your heart rate recently, your blood pressure, anything else that wearable devices can measure, and use that to start studying people and giving them the best medicine. So this is this pharmacogenetics and precision medicine. So precision medicine is really saying, well, we all different. everybody's different. You're all coming from different houses, except for maybe me, because I have an identical twin sister. So we aren't that different, but at the moment we live in different places, so we're exposed to different things, we eat different things. So medicine is personal, and yet it hasn't been in the past. People just say, well, you've got this disease, we'll give you the standard drug. Problem is that some people need twice the standard dose for it to work. It doesn't work in some people. Some people have side effects, other people don't. So the aim of precision medicine, and this is key application of bioinformatics, is getting the correct diagnosis so that you get the right dose, the right drug, for the right indication, for the right patient at the right time. One application of this is pharmacogenetics. So this is basically looking at the genetics of pharmacology. So what changes in your genome are there that might impact how you will respond to a drug? Most drugs only work for 40% of the people. It's most likely because of what population group you come from. Some drugs don't work because there's a genetic change that causes a change in the piece of the cell that's working with the drug or interacts with the drug. So some examples are efavirenz. This is a drug used for HIV. There's a certain change that's in 20% of the population in Zimbabwe that causes, if you've got this change in your genome, you get neuropsychiatric side effects from efavirenz. So if you don't test people in the population and you give it to them, you're causing more harm than you are good from that HIV drug. Aresa is a cancer drug. It only works in 10% of the patients, particularly in Japanese. And the other examples, warfarin is the most common example where there's certain variations, which mean that certain doses are toxic to some people, and for other people, they don't work at all. So you actually have to genotype them, see what their genome says about their warfarin, likely warfarin responses, before you can actually prescribe warfarin in a particular dose. So basically what we're saying with precision medicine is you have a bunch of people who are very different. Now you just give them the same treatment. It'll work in some, it won't work in others. But actually it's better to stratify them, put them into the groups that they are kind of related to each other, and then work out which treatment is best for that group. So it sounds a bit futuristic, but it's happening now. There are very few, there are few cases where it's happening right now. But what will the future look like? So there's a prediction that at the moment we have maybe of the tens of thousands of genomes, sometimes actually a couple of million now, of human genomes that have been sequenced. They're not really in public databases, but they've been sequenced. There's a prediction that by 2030, we'll end up with up to 250 million genomes just for cancer, 36 million genomes just for rare diseases. So we're just talking an unprecedented scale of genomes. Unfortunately, we can't start, we're going to have to start leaving the data where it is. You can't move such big data sources, and also the governments are clamping down. You can't move human data across borders in some countries. So we're having to build new techniques and methods for being able to do federated data analysis, to leave the data where it is and move the compute to the data. How do you kind of manage that when you have the data as private? Is you have researchers that are given sort of researcher passports, and then you're giving a visa to access a particular data set. So is it science fiction that everyone's going to be sequenced at birth, that all your data will be available to the government? You have that updated daily in public databases through your wearable devices, that you can predict your health, that doctors can respond immediately. So we're moving to what we have this P4 medicine: predictive, preventative, personalized, and participatory. Everybody gets to participate in their own health and their own medicine, and you kind of become a doctor and you work with your doctor. Will we sequence everything on Earth? Well, there's already the Earth Biogenome project. This is a project that's aimed to sequence and characterize every eukaryotic type of organism on Earth. That's already started. But the other thing is the scary side of science fiction. What used to be science fiction is now what we're seeing. So movies are becoming reality. These movies about outbreak and contagion. Well, look at where we are now. We're right in those movies, even worse, in fact. So science fiction is not necessarily unheard of. COVID is changing our future, so we're having to do remote working. Luckily, as bioinformaticians, we can do that. Remote schooling, potentially decreased travel. Who knows how long that'll last? Monitoring of people through mobile devices for contact tracing. Will that continue? Will you continue to be monitored by your governments? But the good thing is that it is increasing our cooperation and collaboration. So we have increased collaboration between some governments. I wouldn't say the US and China is a very good example right now, but generally there is increased collaboration between companies, between scientists. There's a huge host genetics project that's an international effort promoting data sharing. So we've got over 16,000 COVID genomes now. The way the scientific community has come together to tackle COVID-19 is something I've never ever seen before. People working together who would never work, have been working together, people sharing data who would have never shared data in the past. And all these portals of free data that are all coming up to drive the science, which is it's truly phenomenal because of the necessity. So we're generally having an increased reliance on managing data sharing, managing big data. Somebody did a comparison with astronomy, YouTube, and Twitter. And I won't go through this in detail because I'm running out of time, but it's very different, the kind of data that we're dealing with in terms of data size, but also complexity and how it's coming in. So we're going to have millions and millions of species just in genomics, never mind the rest of bioinformatics and other omics. The bottleneck's going to be in computing and storage, and encryption and analysis. Yeah, I don't have time to go through this table, but it's quite different to astronomy and YouTube, for example. So we have to deal with this data. In order to do that, we have to embrace the fourth industrial revolution, the 4IR. We have to worry about data integration, mining, visualization, embrace artificial intelligence, and therefore generally grab onto data science. So how is this data science, computational biology, and bioinformatics changing our world? Well, bioinformatics enables us to improve data standards and curation, build tools for these various aspects of data management, but mostly build tools for data integration, interpretation, and application, and of course, visualization. And this needs a combination of data science, computer sciences, life sciences, and you put those groups together, mathematicians, statisticians, you have endless opportunity for amazing real-life applications. I've got computer scientists in my group who could work in the finance industry and earn a lot of money. They work for me and earn a pittance because they love the real-life applications of working with health data, working with things that matter. So bioinformatics has a real potential to have a major impact on health, and with this precision medicine particularly. So, we're starting with the pie chart of little data, lots of information, move to lots of data, little information, but with the help of these bioinformaticians in the middle, these interpreters and data scientists, we can increase our knowledge and therefore work with life scientists to get real applications. We need more computational biologists. Please join up with ISCB and become a computational biologist. So we need skills in data analysis. This is just showing the European data market is billions of dollars in the data market at the moment in terms of staff and all the other aspects of data. One way we've been promoting this is we've got this MGen Africa app, which is a mobile phone app and a tool, and this is for grade 12 learners. And it's to basically, there are various different parts to this, career, live chats and things. And it's to, in the career side to show, well, how have some people gone into genomics and how did their careers develop? So Katala, for example, is from an extremely poor township. She's now got a PhD. She's doing a PhD. She's traveled to international conferences to present her work. You have live chats between people like that and high school learners, and it inspires them to think about how cool genomics and genetics is and to get involved in the field. The web app also provides learning resources, so links to YouTube videos for genomics, brings up ethical issues, so asks the question, ethical questions, because there's a lot of ethics around data and bioinformatics and managing particularly human health data. When should you sequence someone's genome, and when should you pass on results to people? And generally making training fun. So it's got an online quiz, and we've had some 2 quiz competitions where we've teamed up with our Department of Education and many other partners here And this was the 2018 quiz, where you basically had to have all the high schools competing in genomics questions and computational biology. And this is the quiz from 2019. And we also have fun with it, had karaoke about DNA and all sorts of things. And also career talks that get inspired by people like Katala, who I mentioned, and Wisdom, who's in my group from Ghana. Also came from a very small village in Ghana. It's now doing his PhD in Cape Town. And they got to win great prizes. So this lady here is going to be off, was going to be off to Tanzania in September for one of our meetings. Of course, that meeting might not take place anymore because of COVID, but she will get an opportunity to travel. So that is how we're trying to inspire people to stay in, to get into the field. Why did I stay in the field? Well, I love getting people involved. I love, it's rewarding seeing science happen, seeing the students just get engaged with science. It's interesting and fun, apart from all the admin stuff I have to do. Genomics has amazing applications. It's really kind of getting more and more data driven, which is why I've moved away from the wet lab. I haven't dared to pick up a pipette for years. And the interesting thing is that the more that it's becoming data driven, the more they need people like us. So bioinformaticians are just the most in-demand people in the world, I think, at the moment. The field is moving fast, so it's constantly changing, it's exciting, it doesn't get stale. And it can be done from anywhere. So I'm in lockdown at the moment in South Africa, and I'm carrying on almost as normal, apart from sitting online all the time. But it can be done from anywhere. Science is very international. I've had loads of opportunity for travel. These are just some of the countries I've been to only for work. When I say Europe, I've been to probably all the European, Eastern Europe, Western European countries at least. And they're great parts of travel. So you get to do great science, hear about great science, but you also get to visit amazing places, meet amazing people. The Mauritius one, that was our work dinner, was on a yacht in Mauritius, which was not too shabby. So that's my journey. That's where I see bioinformatics. I spoke mostly about the present, but there's just huge potential. And I'm open for questions. And I'm going to stop sharing so I can see the chat.
Thank you so much, Nikki. That was wonderful. We do have a couple questions for you. We have one coming here from Gustavo. It says, about the microbiome, is QIIME the best tool to work with this data, or do we already have something more interesting coming down the line?
Yeah, so Chime is, we used to use that. We've already moved to Data 2, which is a new pipeline. And there's even a new one coming out. I can't remember the name of it, but it's literally evolved. So we started a microbiome project. The first set of data was run on Chime. And already now we've had to move entirely to these new pipelines. And also it depends on the database. So the database, underlying database, reference databases are also changing. So you not only need to keep up with the new tools, but the new databases. So you can, Chime is got quite a different kind of approach to analysis to some of these new tools.
Dr. Mulder, I have another question for you. How has the effect of folding at home increased since COVID-19?
How has the?
Folding at home?
Building at home or working from home?
Well, I think that was a question that was posed there, and I thought.
Sorry, I'm just trying to understand your question.
So the question is, how has the effect of folding at home increased since COVID-19?
Val, did you get that second word? Because I'm just missing that.
Yeah, folding at home, how has it increased since COVID-19?
Folding at home.
I'm thinking folding at home. I'm wondering if it's a program, possibly, folding at home?
No.
We may need a little more clarification on that question. So the person that posed it, if you'd like to just give us a little more data, I'll go ahead and move on to another question. Someone posted the question, Anisha, about, I'm very curious about the role that the heterozygous advantage plays in the diseases you studied. Did your sequencing of the TB genome contribute to the discovery of the heterozygous, sorry, I'm having trouble, heterozygous advantage relationship between CF and TB? If so, how, in general, how can the heterozygous advantage be identified through sequencing?
So what do you mean? So heterozygous means that, so in a human, you have 2 copies of the chromosome. And so if you're heterozygous for something, it means that one copy's got the original and the other one's got the difference. And so homozygous means both of them have got the change. So both of them are the same. So by CFD means cystic fibrosis. So my TB work was not linking at all to other diseases. So it was just, so we were looking at the TB genome. So we look at the TB genome itself, the kind of things we look at in the TB genome, which wouldn't have, heterozygous or homozygous, because it has to be a diploid to have that, would be to look at things in the TB genome that might affect how different strains work differently. In the human host, we have looked at things like are there any genetic changes in the human host that are increasing susceptibility to tuberculosis? And in fact, there are a few that have been discovered, although they differ by different population groups. So if you look at a study in Ghana and a study in South Africa, it's going to be quite different. So that the heterozygous thing is, I don't know what you're talking about, heterozygous advantage, but it's the heterozygous is that if you have one copy of your genome being normal, let's say normal in quotes, and the other copy of that genome having a mutation, then you have to, you've got a 50-50 chance of the right thing happening. And whereas if you're homozygous, you just don't have the right copy. So it's always going to be the changed copy. So I'm not sure if that really answers your question, because I don't know if you meant cystic fibrosis. We haven't looked at any link between TB and cystic fibrosis. Okay, so I'm looking at the Q&A and the questions.
I think there's one left, Dr. Mulder. With the race between the public and private sector, who's won the race?
It was the public sector. So, so there's a lot of politics that went into it. So Eric Lander, who's from the Broad Institute in Boston, he was running the public one, and somebody called Craig Venter, who started the Institute for Genome Research, and is now called. So at that time, he had this company called Celera. And Celera was trying to sequence the genome, and they had all these resources because they were a company, and he was very pioneering in many things. But the one thing I think he was doing wrong was to try to sequence things and then patent them. So what the public did was, as soon as they got a sequence off the machine, they put it into a public database. And as soon as something's in a public database, you can't patent it. So they were able to try and win that race by throwing the things as they came off the machine into the public sector, so that they couldn't then be patented by the private. So the public won. You cannot patent a human genome. You cannot patent even a SNP in a change, a mutation in a human genome, unless it's has a particular application. So you can say, I found this link to a particular disease with a particular therapeutic application. That's when you can patent it. So the short answer is the public one.